talented students, and I've been lucky to work with several very talented students, and two of them are here haunting me. And so I also find this exercise difficult because you want to say lots of nice things about your student, but then you've got other people in the room, and you can't use superlatives because then they wonder, well, what about me? But I can say that Sam has a tell, and I used to see this tell. I'm not going to tell them what it is, so if you do it, they won't know. But I used to see this tell very often, and it was a sign that Sam was nervous. Sam has really exhibited all the best qualities of a PhD student, and she's really joined Carly and Lauren as some of the best examples of what it means to be a PhD student. A PhD is a very difficult exercise. It requires a lot of character, it requires a lot of hard work and dedication, and it really requires personal development in a way that really challenges you as a person. And I've been extremely proud of the journey that Sam's been on, and the way in which she's developed as a person. And I can tell you that I don't see this tell anymore, which tells me that it's time that Sam take over and tell us about what she's been doing. And good luck.
Sounds good. Thank you so much. Excuse me.
Good start.
Thank you so much for that wonderful introduction, Mark. I am thrilled to be here today to tell you guys about two of the chapters in my doctoral thesis. And I've really enjoyed working on this project over the course of about 5 years in the Woods Lab. I particularly find it very interesting and delightfully challenging that I get to combine my love of both chemistry and biology. And I'm super grateful that I found a really strong passion for studying metabolism. And I hope to continue this type of work going forward. And I'm always so excited when I get to tell people why it is I'm putting yeast in an NMR tube. And so,
It would run out of batteries.
excuse me, this was working a second ago. Okay, so I study cellular water exchange, and usually when I tell people that I study water exchange, their mind automatically goes to osmosis. And so on the left and right panels here, I've drawn these stars as some type of ionic stressor. And what this does is it creates an ionic imbalance between the intracellular and the extracellular compartments. And this causes a net flux of water into or out of the cell over a period of seconds to minutes. However, with my research, I assume that the cells are at steady state. And so the cell maintains a constant volume under steady state. However, they can still cycle water rapidly across their membrane. And so a yeast cell can turn over its intracellular volume anywhere from about 2 to 5 times per second. And specifically what I quantify is KIO, and that is the rate constant for steady state cellular water efflux. So KIO is a combination of both passive and active processes. The passive component of KIO is going to be affected by things like temperature, the cell membrane permeability coefficient, and the surface area volume ratio of the cell. Surface area to volume ratio of the cell. So basically, how likely is it for a water molecule to transpire across the membrane? However, there's also an active component to KIO, and that has a lot more to do with the cell's metabolism. And so ATP is the cell's energy currency. And in one experiment, yeast cells were bubbled with oxygen, and under this condition, they can use their mitochondria. And respiration occurs in the mitochondria, and it's the highest yielding process for generating cellular ATP. And under this condition, they saw elevated levels of cellular ATP and also an increase in the rate of water exchange, KIO. However, under the anaerobic condition, cells were unable to use their mitochondria, and they had to use fermentation, which is a much lower yielding process for generating ATP. And they saw a decrease in ATP and a decrease in KIO. And so the active water cycling hypothesis states that cellular ATP levels are directly related to KIO. On top of that, there was a study conducted where they looked at this protein called PMA1. And PMA1 is the most abundant transmembrane protein in yeast, and it has a really important function in the cell. So it will cleave a molecule ATP, and in turn it will eject protons into the extracellular space. And I want to clarify something here, which is that when the cell ejects protons, it's countered by an influx of potassium, which ensures the cell remains at steady state. And so this proton gradient in the extracellular space represents a high potential energy situation where the occlusion of some substrate that is not freely diffusible into the cell can be internalized with the help of the energy stored in this proton gradient. So the collapse of this proton gradient will drive the internalization of the substrate. And it turns out that this happens through secondary transporters, which can co-transport massive stoichiometric quantities of water. And so at steady state, if water comes into the cell, it must, the same volume that came in must also exit, right? So all that being said, water exchange is primarily an active process. It's correlated to ATP and the activity of this PMA1 pump. And so now I want to talk a little bit about how we actually measure water exchange. So many of you will be familiar with magnetic or MRI scans, right? And we can use magnetic resonance to actually quantify KIO. And so in this scan, any area that appears light has a relatively short T1 relaxation time of the water protons. And any area that appears dark has a relatively long T1 relaxation time. And so sometimes what they'll do is they will employ an intravenous contrast agent. And this contains the gadolinium ion, which is paramagnetic. And effectively, it greatly increases the rate of relaxation of the water protons of any surrounding water molecules. And so they inject this intravenously, and it distributes non-specifically throughout the body. And that can be useful when we're looking at something like a tumor, where tumors are known to have sort of leaky vasculature. So this bright tumor here is indicating areas of damage, or where the contrast agent has pooled because the vasculature is damaged, right? But I do want to point out that it truly is telling us about the where the contrast agent pools, right? So there could be other cancerous regions here that are not appearing more brightly because the contrast agent didn't go there. And so I use a similar concept in my research where I'm actually able to add a contrast agent to my cell culture, to my sample prep, and this allows us to see two different populations of water using magnetic resonance. And so this concept is based on something called the NMR shutter speed. So I'm going to show you two images, and if anyone could tell me what they notice is different between these two images.
Blurry.
Yeah, exactly. So this image here on the left is blurry, and this image is more clear. And basically, I had Riley wave pretty fast, and I captured this image with a slow shutter speed, and I captured this image with a fast shutter speed. And so what I want you to remember is that this image has more detail. Now, the shutter speed concept is kind of branching off the concept of the NMR timescale. And so if we look at the NMR timescale in the chemical shift domain, many of you will be familiar with DMF. Now, DMF has a restricted rotation around this carbon-nitrogen bond. And so we would say that this rotation is slow on the NMR timescale, and we end up seeing two peaks for these two different methyl groups. But if we were to increase the temperature of this system, the peaks would resolve due to an increase in that rotation. Sorry, not resolve. You'd see one peak for the two methyl groups because of an increase in rotation. So effectively, we can use temperature as a tool to see more detail in the chemical shift domain. Now, the shutter speed is analogous, but it's sort of looking at this from a different perspective. We would say if we have fast shutter speed, we can see more detail. If we have slow shutter speed, we can see less detail. And so when I measure water exchange, if I were to do an inversion recovery experiment on the NMR to measure the rate of relaxation of water protons, I wouldn't be able to see a difference between these two pools. However, we use the contrast agent as our tool to delineate these two populations of water. And so we have the extracellular water protons, which are exposed to the contrast agent, and they have a short relaxation time, and the intracellular water protons, which have a long relaxation time. And so what we end up seeing is a bi-exponential decay for a fast relaxation population and a slow relaxing population. We can then fit this data to the two-site exchange model because relaxation is occurring in the presence of chemical exchange, and we can yield our values of KIO. And so KIO has been linked to cellular metabolism, and it's thought that it could potentially be a biomarker for metabolic diseases. So I just wanted to give a little bit of background on current imaging techniques that are used. So we have anatomical imaging, and one example of this is a CT scan. CT scans are excellent at spatial resolution. However, if you look at this image here, we can't really distinguish the different tissues in the brain. So sometimes you could use MRI, and MRI has great spatial resolution, but it has superior soft tissue resolution. However, the fundamental issue with an anatomical imaging is that the cell is the most basic unit of pathology and not the tissue, right? So if you want to learn more about what's going on in on a cellular, more of a cellular level, we can use something like a PET scan. So a PET scan employs a radioactive molecule of glucose, and cells that have faster metabolism will actually take up that radio tracer more rapidly, and we can identify metabolic hotspots. However, a PET scan has really poor spatial resolution. So a lot of times what you'll see is bimodal imaging, where you'll do an MRI or a CT combined with a PET scan. But the biggest disadvantage of CT and PET is that they expose patients to ionizing radiation. And this is done in a controlled dose. However, any exposure to ionizing radiation can increase someone's risk of cancer. So the idea here is that MRI is an anatomical imaging technique, but we could potentially make it also a metabolic imaging technique if we can understand what influences KIO. And so before I present this next slide, first of all, I just want to say that I've presented this image so much in group meeting that we affectionately refer to this image here as the map of Australia. So anyway, KIO has actually been measured in cancer patients before. And what they've done here is they took two different cancer patients who had breast tumors, and they imaged they used magnetic resonance imaging to map out the KIO values across those tumors. And what I want you to notice is that there's a high degree of heterogeneity across the tumors here in the KIO values. And tumors are known to be metabolically heterogeneous. And so what they did is they the same experiment after the cancer patients were treated. Now, the patient on the right here had a better or a relatively good cancer outcome because they saw a reduction in tumor size. And what they noticed is the average values for KIO decreased. And the opposite was true for the patient on the left here who had a worse outcome, and they saw elevated values of KIO after treatment. And so this is a really good proof of principle that KIO could potentially be used as a biomarker, but the molecular mechanisms that drive KIO remain incompletely understood. And so the overarching goal of my doctoral work is to characterize the molecular mechanisms that drive KIO. And if you wanted to do this in vivo, it would be very difficult. And that's because what you're looking at here is images comprised of voxels. And a voxel is like a 3D pixel, and it can contain up to 100,000 cells. So if we want to glean meaningful information from KIO, we need to characterize it first on a homogeneous cell line. And so it turns out that yeast cells are actually a great eukaryotic model for this type of work. And to explain why, I first need to explain a little bit of background about metabolism. So typically, human cells will consume glucose. This will get converted to pyruvate, and then it will enter the mitochondria, and where respiration can occur. And respiration is a very high-yielding process for generating ATP. However, it was Otto Warburg who found that cancer cells will actually upregulate fermentation even in the presence of oxygen. And this is interesting because fermentation is such a low-yielding process for making ATP. However, yeast cells will actually exhibit this analogous metabolism. And it was Herbert Crabtree who found that yeast cells will repress respiration under conditions of high glucose. So this is an analogous metabolic regime to the Warburg effect in cancer, which is one of the hallmarks of cancer. But if you give yeast low glucose, they actually will take in glucose and they will undergo respiration preferentially, and that's because they need to be more efficient with their metabolism. And so yeast sort of have this binary metabolism depending on the glucose concentration. So we knew we wanted to carry out these studies in yeast, and there's a couple different ways you can grow yeast in a lab. One way is using batch culture. So batch culture is like the one-pot synthesis version of biology, growing yeast. Okay, so what you do is you add your yeast to a flask that contains a finite amount of nutrients, and they will grow and expend the nutrients over time. And so this is a metabolic profile of cells grown in batch culture over time. And we can see the ethanol concentration spikes, it goes down, the pH will change, and also the rate of proliferation will change over time. And so what we came up with is, instead of doing batch culture, we wanted to grow our yeast in chemostat bioreactors. And I have a love-hate relationship with chemostats because it took me so long to get them going, but it was totally worth it, and I'll tell you why. So what happens here is we have these glass tubes, which Josh made in the stockroom, thank you very much. And we add media into our cell culture at a set rate, and this is controlled with a pump. And then we bubble the cells with breathing air, which sort of pressurizes the vessel, and it causes the rate of efflux to be exactly equal to the rate of media influx. And over time, the cells will actually adapt to this nutrient flux, and they will maintain a constant metabolic profile, a constant rate of proliferation. So if I were to make a graph like this for the chemostat, it would look like straight lines no matter which time we sampled. So it's ideal for studying metabolism. So we sort of wanted to go back to this idea that yeast have this binary metabolism depending on low glucose or high glucose conditions. So I've coded this as purple being low glucose and blue being high glucose. So looking at the metabolic profiles of these two metabolic regimes, we found that the cells given high glucose took up more glucose. We also found that they grew to higher cell densities. But the most striking difference between these two metabolic regimes is the absence of ethanol in the cells given low glucose. So that tells us that they were not doing fermentation. They were likely doing complete oxidative phosphorylation, which is just using their mitochondria. And so the cells given high glucose did indeed have ethanol in their exometabolome. And another notable difference between these two regimes is that the pH was consistently lower for the cells given high glucose. All right, so of course we want to get down to business and measure the rate of water exchange. Oh, excuse me, before I move on, I just wanted to say that I hope I've convinced you that we are able to have cells that either are respiring or fermenting, depending on glucose concentration. And upon further literature search, we can anticipate different morphology of these cells as well. So these are electron microscope images of yeast mitochondria, and we can see that the respiring yeast have very small and many mitochondria, and vice versa for the fermenting yeast. So from now on, I'm just going to call the high glucose fed cells fermenting and the low glucose fed cells respiring. Okay, so then of course we wanted to measure water exchange. But to do this, we also needed to measure cellular ATP levels. And that's because the active water cycling hypothesis says that KIO values are correlated with cellular ATP levels. But this brought us to a fundamental question, which is the respiring cells were fed low glucose, but they have a very efficient metabolism. Whereas the fermenting cells were fed 7.5 times more glucose, but they have an inefficient metabolism. So which culture was producing more ATP? Well, we hypothesized that the respiring cells would produce more ATP and that their water exchange levels would be higher than for the fermenting cells. And so to carry out these experiments, I would grow cells in the chemostat steady state. I would then harvest the cells and add a extracellular contrast agent. And then I would do my magnetic resonance techniques to yield the values of KIO. And so to measure ATP, we went ahead and we got this commercially available assay. And what it does is it reacts with ATP to produce light. And we can actually quantify the amount of light by comparing our samples to a standard curve of just ATP and the assay. And basically what this assay does is it has a firefly enzyme, right? So it reacts with ATP to produce light. Okay, so we first wanted to measure KIO, and this is our result. We looked at a couple of different strains. So we have baking yeast, we have FY4 and BY4743. I know it sounds like an alien, but these are just lab strains of yeast. And then we also looked at some yeasts that were genetically modified. And basically they had reduced ability to produce certain proteins, right? But what I want you to notice here is that across the board, the respiring cultures of yeast had faster rates of water exchange, whereas the fermenting cultures of yeast had lower rates of water exchange. And so, of course, then we had to go and measure ATP. And I found that, looking here at the percentage of ATP per cell, I normalized the respiring cell's ATP value to 100, and I saw that the KIO values and ATP were correlated between the two metabolic regimes. Now, except for this result that I got. Now, PMA1 had the same approximate amounts of ATP per cell, whether respiring or fermenting. But this is a little bit of an outlier, and I'm going to try my best to explain why. So, PMA1 is really important under conditions of high glucose. And it also happens to consume up to 60% of the cell's ATP under conditions of high glucose. And so we would expect that while fermenting, less of the protein was available to hydrolyze ATP, leading to elevated ATP levels under the fermenting regime. So aside from this outlier here, we can say that we're able to delineate different cultures of yeast based on their KIO value, which could have a profound impact on the way we look at KIO as a biomarker. And so this got us thinking, if the respiring cells have these elevated KIO values and they are dependent on their mitochondria, do mitochondria directly play a role in the rate of water exchange? And so we wanted to go ahead and use the cells that I've been calling fermenting as our model for these experiments. But there's something I need to clarify here. And that's that if these fermenting cells were completely consuming glucose and fermenting all of it into ethanol, we would expect to see a 2-to-1 ratio of ethanol produced to glucose consumed, because it's a stoichiometric process. However, what we see here is actually a 1-to-1 ratio, approximately 1-to-1 ratio, it's not 2-to-1. And so we can presume that those cells are likely undergoing a mixed metabolism, where they're doing fermentation, but they're also doing respiration at the same time. And so knowing that, we wanted to go ahead and target the mitochondria in these cells. So how we do that is we would grow our cells in the chemostat, harvest them, then we'd add this mitochondrial modulator called 2,4-DNP, and we would measure the ATP and the KIO values with increasing concentrations of that drug. And so we hypothesized that if we mess up the mitochondria and we would see a decrease in ATP levels and also KIO values. Now, I want to explain to you how this drug actually works. But to do that, I first need to talk a little bit about how the mitochondria works. And so I want to make it abundantly clear that we are looking not at the cell membrane here, but we're looking at an organelle which is inside of the cell. And this is my attempt at drawing a mitochondria. So they have an outer membrane, they have an inner membrane, and they have this intermembrane space. And what happens is cellular metabolism results in protons being pumped into this intermembrane space. And this represents a high potential energy situation. Now, the majority of these protons are confined to this intermembrane space, but as a little bit of an aside here, mammals will actually allow some of their protons to leak out, and that's how warm-blooded animals actually stay warm, because this energy is released as heat. And so it turns out that sea otters have one of the highest rates of mitochondrial leak because they live in cold water, right? And so they need to stay warm. Okay, but going back to the mitochondria here. So these protons represent high potential energy, and the only way they can escape the intermembrane space is through this protein called ATP synthase. Now, the energy stored here is not useful to the cells, so the cell needs to make ATP. So what it does is it utilizes the collapse of this proton gradient in order to catalyze the reaction of ADP to ATP. And ATP synthase is not a stoichiometric enzyme, but it is a mechanical enzyme. So it's sort of like a wind turbine, where the more energy you have stored in this intermembrane space, the greater ability you're going to have to produce useful energy to the cell, which is ATP. And so what DNP does is it's a small molecule, and it can cross the membrane in both acid and conjugate base forms. And what happens is it will become protonated in this intermembrane space, and it will diffuse across the membrane, which disrupts the proton gradient, which should theoretically lead to a reduction in ATP. So I went to the lab, and I first wanted to add DNP and measure the effect of DNP on ATP. And I thought it was going to be so easy, and that I would see a reduction in ATP. And so I went to the lab, and I first got this result, where ATP per cell is plotted here, and 0 DNP is normalized to 100% ATP per cell. And I saw an actual increase in ATP. And I was like, oh, that's weird. I guess I should just try it again. And so I tried it again, and I got the same result. Now at this point, I was really questioning my life. I was like, what am I doing? How could this be possible? But if I've learned anything in grad school, it's that if you get the same result twice, it's telling you something. And so I went into the literature, and I found a lot of interesting history about DNP. And so DNP was used in munitions factories in World War I in combination with picric acid. And it was reported that the women working in these factories would be really skinny, that they would develop cataracts, they would turn yellow due to prolonged exposure to DNP, which is yellow. And one of the workers, the first death, was reported due to hyperthermia, which is overheating. And so Maurice Tainter studied the effect of DNP on cells in Stanford University, and he found that DNP can make your mitochondria less efficient. So it causes mitochondria to leak. And remember, those leaky protons are released as heat. So that explains why the workers were overheating. And so because of the correlation to DNP and weight loss, it was actually marketed as a diet pill. But it was very shortly after taken off the market because of its really narrow therapeutic window. So at high doses, it could be very toxic and it could lead to cataracts and death. But it's actually emerged recently as a popular illegal supplement with bodybuilders, which has prompted studies on the safety of this drug and also public health warnings not to take it. And so, thinking about my results, it all started to click when I started to learn about this. And I found out that what happens is your cells are not, they're not just going to sit around and die, okay? They are dynamic entities and they want to stay alive, right? And there's tons of feedback systems. So when this proton gradient gets disrupted, the cell will actually sense that. And in turn, it will increase its metabolism, which could potentially result in an increase in ATP, okay? And so I went back into the lab and I added DNP to 2 other strains, and I was able to confirm that the result was not strain specific and that I saw an increase in ATP in 2 other strains as well. So then I wanted to go back to the first strain, and what I did is I greatly expanded the range of DNP that we were looking at. And so what I found was that ATP would increase with increasing concentrations of DNP to a certain point, and then it starts to decrease. And this result, this type of curve is called a hormetic response. When I learned this word hormetic response, everything's just, my world changed basically, because I was like, this makes so much sense. So basically you have some drug that you're adding and you get a positive response at low doses, and then you get a negative response at high doses, which is exactly what we're seeing here. So think of something like caffeine, where if you have your morning cup of coffee, you're feeling great. If you have 20 cups of coffee, you're not gonna be feeling too good, right? And so then I went and I measured KIO. And I was really happy to see that the KIO values mapped almost exactly on this ATP curve, which supports the active water cycling hypothesis. And it also provides a really important control experiment, because we saw KIO values increasing and then trending decrease as well with decreasing ATP. I was also able to confirm that when I added DNP to these strains here, we saw an increase in KIO. And so just to summarize, we saw this hormetic response, and I think that the way we grow our cells, they are particularly prone to exhibiting this hormetic response. And that's because when we add DNP and it indirectly causes metabolic stimulation, the cell will take up glucose. And at steady state, these cells have an abundance of glucose in their exometabolome, so it's readily available for them. They also have this mixed metabolism, which I talked about, where they can generate ATP not only from their mitochondria, but also from fermentation. And so at high DNP, the cells just can't compensate for the presence of that inhibitor, and we see a decrease in ATP and a decrease in the rate of water exchange. And so what we did here is we targeted something that was on the inside of the cell. And we saw changes in water exchange, which are usually thought to occur at the cell membrane, right? And so that could have a really interesting impact on the use of KIO as a potential biomarker. And so, just to conclude here, we showed that chemostats are really great for studying metabolism and to characterize the molecular mechanisms that drive KIO. We showed that cell culture can actually be differentiated based on their KIO values. All of our results here fell in line really well with the active water cycling hypothesis, and that intracellular activity could potentially affect KIO, which warrants further study, and it shows that KIO can be very useful as a biomarker. And there's other metabolic diseases and disorders that could be looked at for KIO to be used, and it could be useful. And so with that, I just need to thank a couple of people, and I really have to thank my lab. They have been my biggest support system. I'm really grateful Lauren and Carly are here today because they are my inspiration, women in science. I've always looked up to them. And then Abigail and Charlene have been just best friends you can imagine, just great people. Mark, of course, has been a great advisor, and I really appreciate him challenging me to become a better researcher. And I have to give a special shout out to Joe Armstrong and Jonathan Dutra for helping me do this work, and Abigail, who's going to be picking it up. So I would love to thank my committee members for being a great support system and for the productive conversations we've had over the course of these 5 years. And then collaborations with Dr. Thompson for helping me with flow cytometry, and Dr. Bajrowski for letting me use his plate reader and his students as well. The office staff have been instrumental, amazing people to work with, just wonderful, amazing people. The stock room, they are so helpful. Thank you guys so much for everything. I really appreciate working with them, and I'm glad it's them and not anyone else. I have to say thank you to my family. I'll try not to cry, but I didn't just fall out of a coconut tree. It turns out I exist in the context of everything that came before me. So my mom and dad are just, I'm so inspired by them. They are amazing, and they've been, excuse me. I'm sorry. I'm sorry. Okay. My sisters, my family. My sister Jasmine just got married, so that's really exciting. And then there's so many grad students in here right now, and that just means, excuse me. My friends, thank you for being here. I could not have done it without you. Sorry, I need to think about dead puppies or something. No, my friends are so inspirational and motivating, and I really, really appreciate the chemistry department and the support I've got. And thank you so much for listening.